Dr. Pen, shall we?
Yes.
All right. Good morning, Doctor and delegates. Good morning and a warm welcome to this online symposium on a Sunday morning. We sincerely appreciate each of you for joining us today. Well, today, as you're aware, today's symposium is related to the science of aging reversal, which is young plasma conditioning and cell viability tests in heterochronic parabiosis. We have actually organized and conducting symposiums related to this topic since the year 2019. A physical symposium before the COVID-19 lockdown, typically in Kuala Lumpur and Penang. And also online webinar ever since the pandemic struck us. Therefore, yes, we are committed into this heterochronic parabiosis project. And things has been changes in terms of our advancement in technology and our approaches throughout the pandemic lockdown. And now we see an even better potential in its the potential to expand a human health span. So we are honored to have Dr. Pan as our distinguished speaker today. Dr. Pan graduated from NTU, Taiwan National University, and hold a PhD in chemistry from University of Chicago, along with a medical degree later. As many of you are familiar with Dr. Pan's exceptional expertise, so we won't go into further details about his background. Anyway, thank you all for your present support and enthusiasm. Let's make the most of this opportunity to deepen our understanding together. And please jot down if you have any question during today's webinar, please jot down your question and we will have a Q&A session after the Dr. Pan's sharing stand. And I would like to invite you to turn on your video as well, to create like a better atmosphere online. Okay? So without further ado, let's extend a warm round of applause to Dr. Pan for his talk today. Dr. Pan.
Okay. Thank you, everybody. Thank you, Suho, for the kind introduction. And I again thank you for the time in the Sunday morning, and we get together online and to try to learn and study some more about the technologies in aging reversal, and of which I have been involved for almost 29 years now. Anyway, the title of today's talk is Young Plasma Conditioning. And we are going to spend a little bit more time and discussing about the stem cell viability test, which is very important for the cell therapy and as well as for the health span in longevity issues. And the technology is based on a 100-year-old experiment, which is called heterochronic parabiosis. And like Mr. Lu just said, my background is a little bit awkward. My original PhD was in physical chemistry. And after that, I was recruited by the US government into the medical scientist program in the 1986. So the way I look at the medical problem may be a little bit different from most of people who practice more classic medicines. And another thing is, by professional, I'm boarded by the American Board of Plastic Surgery. So what we are trying to do is probably is a little bit more of a surgical approach rather than a medical small molecule medicine type of approach. So that will be a quick introduction about the way we are going to approach this subject. And these are some of the disclosures, and you can see that we have been around for about 20 years all over the Asia. And in fact, I'm giving this talk in our office in the Shenzhen, which is the third largest city in China, and the population here is 23 million. And this is our own new clinic, and we collaborate with the local partner and set up the clinic last year, and we are practicing the plasma conditioning therapy here. And the talk, I organized my talk like as the following is, we talk briefly about the background and the hallmarks of aging. And since most of you are professionals, so we will go through this very quickly. Then we go through again very quickly on the history of the heterochronic parabiosis, which I think is still important, not to forget all the giants and who let us stand on their shoulders. Then we talk about the clinical application of the heterochronic parabiosis and the young plasma conditioning program. And here we are going to talk to you more about how do you interpret the stem cell viability test. And I think this will be the key element in terms of the success for the practice of young plasma conditioning. So as you know, I mean, aging is in proportional to the chronic disease. And I think this goes without saying. And most of people after age of 60 is going to have at least 1 or 2 chronic disease. And by the age of 80s, then it's very likely they're going to have 5, 6, or even 8 chronic disease. So the concept of the medicine has been changed dramatically after the population aging has hit many developed societies like a tsunami. So the concept now is not like the traditional medicine, which is increase the lifespan. What is more important now and what most people are eyeing at is more like the increasing the health span. So in other words, that what people are looking for is more like living healthily with as few chronic disease as possible and with as little disabled time before their death, okay? And so that's the concept of the health span rather than the lifespan. So we don't talk about longevity all that much anymore because we see so many people lying in the senior home care facilities and who had a stroke, who had cancer or heart failure and lying there for 8 or 10 years and before they die. And I don't think that's the life that most of people want. So what we are trying to address is actually aging-related disease. So since this has gained so much attention, so scientists and the doctors all over the world has proposed many mechanisms, and what's listed on the left side. And I think most of you, since you are professionals, I'm sure you are familiar with at least some of these entities. And in terms of the logic, the causal issues of all these mechanisms, you can summarize in many different forms. And this is one of the interpretation we're trying to address the aging-related disease. But regardless of which principles do you believe in, but I think as a whole, it's still a very complex and messy situation in terms of our understanding of the aging and the chronic disease. So like the Judith Carpini, and she was one of the first lady who proposed the idea about the hallmarks of aging. She always said the dirty nature of biology. And since we are medical doctors, so unfortunately, we are facing the dirty nature of these related issues. And what makes things worse in terms of the translation of all the findings in the laboratory to the clinical practice is very low. Based on the most recent FDA data, there are only 8% of all the published and proposed clinical trials who finally makes it to the market. So as a basic scientist, which like what I just said in the beginning, I'm a chemist, and in particular, I'm actually a physical chemist. So we would like to ask ourselves the first question: That is there any basic science in aging? In terms of basic science, is because it is more repeatable and is quite different from the biological and the social science. So the first approach we took back in 1994 was looking at the genetic versus epigenetic principles. And what, at that time, I was teaching in University of Pennsylvania. And the first epigenetic factors we look into is actually the chewing of the nuts, the effect of chewing nuts on the form and shape of the masseteric muscle, okay? And that was a big, has a very important factor in terms of the craniofacial surgery. So, but in terms of the aging-related chronic disease, we need to address a little bit different issues. So the first question we asked after we're trying to address the aging issues with a basic science approach is the cellular aging or the aging itself is programmed. That means that if you are genetically, you're going to be have a long life, and that's programmed. So regardless of what you do, you're going to live for a long time. Or if you have a gene which could make you have a short life gene, and so it doesn't matter how well you take care of yourself, the cell is going to die anyway. So it's a pretty good experiment that was done by a German group. And what they took in this slide, on the right lower quadrant, you can see this old age group. They took the MSC, mesenchymal stem cells from the hip and the bone marrow of the hip of people over 80 years old. And they put them into a growth media, which is rich in the standard growth media, and try to measure their cumulative population doubling time, okay? So you can see that if in some of the cases, the old cells or the cells from the old people actually outgrow the cells from the younger individuals. So this experiment tells us circulating progenitor cells, that's the other way you can look at the stem cell. The levels do not decline with healthy aging. But if you have a trauma or you have a disease process when you were younger, because of the change of the disease on the environment in your body, actually that will eventually lead to decrease in the progenitor cells. So the answer to this question is not entirely. At least we know if you're giving the right cultural media or environment, the cells from the old people can actually behave just as good as the cell from the young counterpart. So the first conclusion we have is aging is at least partly due to the environmental factors. So, I mean, you probably, if you are not familiar with this concept, and I can tell you two things, which is two very popular concepts in terms of anti-aging are directly related to this concept. One is the diet concept. So the calorie restriction and all the different Mediterranean diet and the Atkins diets, all these things are actually intervention trying to alter the systemic menus. So this belongs to an epigenetic approach. And the other one, of course, is the exercise. And the data shown that with adequate amount of exercise, you can prolong the lifespan, and together with other measures such as quit smoking and the stress management, things like that. So all those methods, which I just mentioned in the last 15 seconds, shares one common characteristics. That is, they are not very expensive. In fact, many of them are actually free. You can run on the street freely, okay? But these free issues are usually very difficult to last. So as a result, you can see people on the streets of the America in Europe, that people probably has been doing exercise and dieting since their teen age until they're growing old. So, and the issue, you see this phenomena is because this requires very, very high discipline, and it's very difficult for average people or average people, people to manage. Okay. So, is there any other ways we can do this other than the stress management, other than the exercise, other than the diet, calorie restriction, and things like that? Yes. Okay. And I think most of you probably see this news. This was just about 10 days ago on the news, okay? In fact, it's on the May 22nd. So this news is, I think, hit the all the social media very, very hard. So many of at least 10 of my friends has sent me a link to this news. So basically what it says is one guy, the guy on the right side, and Mr. Johnson, and he is a rich people by selling his online financing company to some big tycoons. So he get 800 million cash back. Then he's begin to spend about $2 million, he claims, every year to retain usefulness of his body. So this guy has been on the very active in the social media and very been reported for many times already. But this time he took one step further. Okay, he asked his son, which is shown on the left side of this picture, and to donate blood for him. And what makes it even more interesting and gets people's attention is this time he actually donated his blood to his father. So as a result, this is like a 3-generation swapping of the blood. So this one has definitely caught the attention of the media. But in fact, this what the place that did their plasma exchange, the concept is not a new one. It's a company. I'm sorry, I have to say that the company is a bit controversial, but I think it's a very talented and innovative company. And the company called Ambrosia, which is established by a Stanford Medical School graduate. His name is called J.C. Kamazin. And since 2014, he has been managing to collect blood from the college students in the Silicon Valley area and resell it to the tycoons in the Silicon Valley area. And the cost for that is about 1 liter of college student blood, and it's going to cost them about $8,000. That's the price. And the company has been around, and I think they are still active right now. So like all the new technology in medicine, the FDA and the conservative media and everybody has to come out to say something. Safety is #1 concern and things like that, just like you can't get on the airplane, okay? So, and this is a comment which I clipped from the news report. So basically, all what it says, first thing is frequent donation does have positive effects. Of course, otherwise nobody wants to donate blood. A lot few people will quit. Okay, so the next thing is plasma swapping. The result of plasma swapping in increasing longevity is inconclusive. It doesn't mean it's wrong. Okay, it's only say it's not conclusive. Okay, and the next thing is a viable human. Is this a viable human treatment? Is not enough. Not enough. It doesn't mean nothing. Okay, not enough. The last thing is to him, the guy, Charles Brenner, is a biochemist, and in the City of Hope, he says to he himself is gross evidence free and relatively dangerous. So with all those comments and some supporting comments from the Red Cross, and I want to accept the target for today's talk is that we'll try to address this skeptical and the comments about the plasma swapping or plasma reconditioning and things like that. And we will try to provide more evidence and trying to see whether trying to make these things more conclusive, and maybe we can raise the evidence level to convince people who is skeptical about this technology. So the concept of heterochronic parabiosis was started in 1864. That's 1.5 century ago. Okay, and it was started in France. Okay, by a guy called Paul Bert, and he's the guy who connect the two animals, two rats, two mice usually. Okay, one animal and the older animal, and this technology was attempted again by the genius surgeon called Alexis Carrel, who was my alma mater in University of Chicago. But the guy who bring the heterochronic parabiotic technology for the study of aging is actually in 1957 by Clive McKay. He is also the guy who began to study the effect of calorie restriction. So this is a name to remember, and he is a PhD in nutrition. Okay. So in one sentence, the heterochronic parabiosis experimental technology is the only rapid and visible aging reversal method as of today. Okay. There are other methods like dieting and exercising and things like that, but they all take a long time for you to see the effect. And sometimes you can only see them in terms of the biologically, you cannot see them grossly. I mean, what is different is heterochronic parabiosis. You can see the animal actually just macroscopically just become younger. So what happened is two animals, after they are connected, so they just share the blood because the organs of each animal remains inside of the animal. So the only thing which has been exchanged in between the mice or rat is only the blood. And how do you connect the humans? That's the next question. So we all familiar with the Siamese twins, but these are usually, they are not heterochronic. So this is not a good example for heterochronic parabiosis. That means a different age, okay? So the next one is the pregnancy. So of course, the mother of the fetus is always older than the fetus. So this is the heterochronic parabiosis. But since the size difference are quite big, so the effect of this can be skeptical, and we will talk about this later. But this is pregnancy is the most human model for heterochronic parabiosis, and we will do some calculation in terms of the effect of the pregnancy on the longevity of the mother in the later part of this talk. Of course, you can get pregnant if you are a lady and if you are still fertile, but if you are not, then you probably want to know how can we apply the concept of heterochronic parabiosis separately, okay, without connecting the two persons together. So the concept, the intuitive approach is very simple. You just think that there must be some good stuff in the young blood, and if you adding some of those young stuff into the old blood, you are going to make the old blood younger. And as the result of the younger blood, and the body will become younger. That's the logic behind this. So in terms of clinical approach, this is the old animal and a young animal, and they become older. Then how do you make it become younger again? And this is the concept behind this approach, which was taken by Alexis Carrel back in the early 20th centuries. So what he did is he took a chicken heart and give the chicken heart with broth taken from the chicken embryo. And he was able to keep the chicken heart beating for several decades. And this is the classic experiment of using the heterochronic parabiotic principle in terms of organ preservation. So who took this to clinical level is a famous guy called Dr. Paul Nehan. And he was inspired by the Alexis Carrel's experiment. So what he did, in some coincidence, he was able to begin to inject the tissue extracts from a tissue lamp and to all these celebrities, and which you're all familiar. And what's even more interesting is that the technology later become the famous brand called La Prairie, which is if you travel in the airport, you can always see them. And in the other part of the world, there will be the Japanese. So Paul Nehan started in 1931, and the Japan, they have a company called Lanek and the Melson. They started in the 1970s. So they begin to sell tissue extracts from human placentas. And to date, they are still doing the same things. And a lot of people will travel to Nagoya and I think it's this very famous clinic called Morishita. And they do a lot of injection of these placenta extracts for longevity and the chronic disease treatment. So all these technologies, they are not convincing, or probably FDA is still not happy with them. It's because they are lacking clinical data. And so I think my personal opinion is that the concept or the practice of heterochronic parabiosis was a change, has a paradigm change. And that was back in the 2016. A group in Berkeley, led by Irina Comboy and his husband, Michael Comboy, they did an experiment, okay? So what they do is this. They, instead of connecting the two animals together, they are just drawing blood from the old animal and inject it into the young animal and vice versa. So originally, during the experiment, they were expecting that since you're only injecting a small part of the blood, to say 1/10 of the whole blood, so they're expecting the young animal will get a little bit older and the old animal will become much younger, because old animal has little useful substance. And if you give 10% of the young animal's useful substance, then that can compose a big substantial part of the old plasma. But the experimental results were quite surprising. That was the young animal, it did not get a little bit older. The animal actually gets much older instead of getting a little bit older. And the old animal did not get much younger. The old animal only get little younger. And this seems to be a tragic, very bad result for whoever believes in the useful material. So what the Comboy find is, instead of the concept of young people has more useful substance, it's actually we should switch the concept to the old people has a lot of old substances, and of the young substance and the useful substance, the aging substance actually is more dominant. So this is like when you are driving, and if you have the brake step down, then it doesn't matter how much gas you are pressing, the car is not going to move. So with this new observation, and the Comboy quickly working with the pioneer guy in in plasmapheresis. His name is called Dr. Dobrik Kipro. He's from Yugoslavia and practice this type of technologies in the Bay Area right now. So the two people get together and introduce the concept of using the TPE machine to remove the bad substance and replace the removed substance with the FFP, fresh frozen plasma, or with the 5% albumin and with a certain percentage of the IVIG. And the team keep on working on this, and eventually they were able to do a small clinical trial of I think a dozen patients, and they were able to see significant decrease in terms of the C-reactive protein and other indexes in the blood. The issues is then require doing weekly plasmapheresis for say like 6 or 10 times, and before they stretch the time intervals. So the other company, which is called Grifols, which is one of the largest company for human plasma products, and the group is more disease focused, so they use the similar technology to address the Alzheimer's disease. So the group took the group about 10 years and to complete a level 3A clinical trial. And the results shows that if for Alzheimer's people, the MMSE score, that's the score very commonly used to address the severity of the deterioration of the Alzheimer's disease. The rate of the decrease in terms of MMSE score will be decreased by 70% if you are treated with this TPE technology, therapeutic plasma exchange technology. So on the x-axis, you can see all these little triangles. So each triangle in the beginning is 1 week apart. So you need to do a full volume TPE consecutively for 6 times. Then you can do this, become like every several weeks of low volume plasma exchange, and to gain this type of results. And with that data, the group begin to set up Alzheimer's treatment centers in Europe. This one shows their centers, their first center in the Barcelona, Spain, okay? And the center is designated for the treatment of Alzheimer's disease. So what we can summarize what we know about the clinical application of heterochronic parabiosis from Paul Nienhen, the Japanese placenta extract, and to various forms of plasmapheresis. So we have been involved in regenerative medicine for a long time, but we got into this plasma-related anti-aging methods in the year 2016. At that time, by coincidence, I was brought to Phnom Penh, which is the capital of Cambodia, to address some health issues to one of the generals in that country. And at that time, we were surprised to find that Cambodia has the youngest population in the world because of the red Khmer regions years. So with these so many young blood available to us, so we quickly get into the concept of thinking about perform Ambrosius approach by using 15-year-old fresh frozen plasma. So that was our original thinking in 2016. So at that time, the convoy published their landmark paper about the aging substance dominant paper. So it's not really too hard for us to incorporate the concept of TPE into our 15-year-old FFP concept. So what is that our first designs was trying to use the TPE machine to remove the aging substance and replace them with fresh frozen plasma of the 15 years old. I mean, that was the ideal situation, and that was what we were planning to do, okay? So this is the first procedure which was done in my clinic in Taiwan. Okay, this is again, you can see this is one of the TPE machines, and this is one of the, our patients, and you can see this is a technician and there are two nurses there, and everybody is quite busy trying to do this. Okay, so we quickly find out that to perform the TPE machine, the first thing is that it's labor intensive. Second thing is that I mean, the patient is actually very frightened during the procedure because he later tell me he's not very comfortable of seeing his blood circulating outside of his body and things like that. So for psychological issues and for financial issues, and the most important of all, the TPO is still has a low risk, some low risk of stroke, for complication. So we decided to back down on this approach. So as a chemist, we quickly redesigned the process of clinical heterochronic parabiosis. So instead of using the TPE machine, we decided to use the bloodletting. Since the plasma, the blood volume in the human is approximately the body weight divided by 13, then the blood bank allows us to remove 500 cc of blood each time. So by simple calculation, I think by doing bloodletting, should be able to remove at least 5 to 10% of the bad substance in the blood. And the other modification is that instead of using the FFP of 15-year-old, we begin to using our knowledge about hormone, about cytokines, and the artificial plasma replacement fluid. And we use this to design our formula for the replacement of the removed human blood. So we named this intermittent plasma conditioning because this is by definition is really not a plasma exchange. And we don't use this continuously. We do the conditioning on a bi-weekly basis. So at that time, the Stanford professor called Tony Whisker, and he did a very comprehensive study about the proteins in the blood of different age. So what he studied is about 7,000 proteins in about 3,000 subjects. And with his study, he was able to classify the proteins in the plasma into 4 categories. That are the common factors, means that the concentration between the young and the old are essentially the same. The adolescent factors, that's the hormones related to the adolescence. Most of them are sex hormones. The other one is the beneficial factors, which decrease with age. And lastly, the harmful factors, which begin to accumulate after age of around 40. And that was his finding. And although there are 3,000 proteins in the plasma, and he find out that the candidates for this plasma conditioning is really not that many. So with his data, and we are able to design a formula using a machine to compound it to become our anti-aging plasma replacement. With that, between the year 2018, the third quarter of 2018, until the first quarter of 2020, we performed about 200 sessions, 200 plus sessions in Taiwan and Malaysia and Penang, Kuala Lumpur and Penang. And we don't do this all that smoothly. Frequently, we do encounter allergies, and sometimes we have patients become anemic, And we also have one incidence of cytokine storms. So the interesting part is, after the event of cytokine storm, we were almost ready to quit this project. But one thing which is very important is all these customers, although despite all these allergic reactions and even the cytokine storms, and the customers actually give us very positive feedbacks. So we re-examine what we have done, and we find out we are missing one important piece of all with all this program, like the others. That is, we are lacking a treatment guideline. And it's very interesting in 2020, and we have one major investor become interested in our technology and practice. And he also requests us to begin to provide some hard data. And we use the investor's blood to perform the first plasma examinations in the April of 17th of 2020. And this was his result. Okay. Now, let me explain this plasma diagnostic technology, or you can also call it stem cell viability testing in more details with this figure. So on the x-axis, what you see, the percentage is the percentage of the human plasma. So the on the right-hand side, 100% means that the 100% human plasma from the subject. And we were giving a certain number, say like 1,000 stem cell. And the stem cell will be cultured by using 100% plasma. And we will count the number of the stem cells survived after 1 week. And as you can see on this figure, basically all of the stem cells died up in the human 100% human plasma in 1 week. But if we begin to add in some formulations, we compounded based on Tony Whisker data. And you can see after we compounded with 65% of the human plasma and 35% of the formulation, you can see this mixture will be able to maintain the survival of the original stem cells. And keep on doing so, we find interesting phenomena. That is, with our formula and the combination of our formula with the human plasma, you can actually improve, you have room to improve the survival rate of the stem cells by hundreds and sometimes even up to 1,000 times. And with this observation, we quickly repeated after the test for each individual sessions. So with this, for these individuals, these are the tests from the April 29th and 1 month later. So in between, this patient has 2 sessions of the plasma conditioning. And you can see the viability of the stem cell begin to increase very significantly. So the next question will be, how long will this observation last? So what we did is, after we did 2 sessions for the plasma conditioning, then we waited for 10 weeks. Then we make another adjustment, another measurement. Then we can see that the improvement of the stem cell viability in the human plasma, or these synergistic graphics, actually can maintain quite well. And by keep on doing so, you can come up with a graphic like this. So on the x-axis are the time, and the green circles are the time he got the treatment. So with 6 treatment, with actually 5 treatment, the viability of the stem cell rises from 7% to 837%. and that is an increase of 120 times. And what's even more interesting is, despite of the changes in the plasma diagnostics, the patient's EGFR, and this is shown in the gray dots, okay, that was the original reason he came to us, because of the deterioration of the EGFR. And this triangular after the EGFR after he got the plasma conditioning. And you can see the EGFR immediately stabilized after the couple plasma conditioning sessions. And the guy also began to grow out black hairs. And what's more interesting is that the guy come back in about 3 months later, and you can see the stem cell viability actually dropped to about half from the maximum. So in other words, the effect of the plasma conditioning after you reach the 120 times were able to maintain for about 3 months. So, and you can keep on doing this type of things. You can see the stem cell viability testing results, the correlation with the plasma conditioning is extremely well. And these are the EGFR data. Keep on doing so. So what we can conclude here is that are the following. One is the old blood does not support the MSC, and the conditioning takes 3 to 6 sessions. And although the plasma conditioning, the percentage is probably 10–15%, but the increase in the stem cell viability is by multiple times. Okay, so the results, of course, is patient dependent, but to our experience, most of the patients, up to 90% of the patients, after they did 3 to 6 treatments, you can definitely observe the increase of the stem cell viability in the plasma of the patient. So since all the material we use are not small molecule drugs, not pharmacological substances, so we name this as a biochemical type of plasma conditioning. That means that all the material we use, like nutrients, hormones, and cytokines, all of these chemicals are existing in the patient's human body. And we are just using these biochemical reagents to adjust or titrate their concentrations to reach these results. So to summarize what we see on the previous example, you can see the patients, if you take patient's plasma and give it to 100 stem cells, after 7 days, you originally only have 3 cells survived. And after several sessions of this, the survival rate you give it to 100 stem cell, the survival rate, the stem cell actually can grow to 360. And 6 months later, and the stem cell still, the effect of the plasma conditioning on the plasma still remains. So I think most of you are doctors, and you are aware of that most of the medication, the half-life or the are quite slow, from hours to days at most. So we are thinking by what we are doing is actually we are creating an epigenetic type of vaccine for aging. Okay. So I know some of you do practice albumin therapy for anti-aging. So let me see, we did do an experiment using the albumin and use that to compare to our data. So on the left is the result of the intermittent plasma conditioning, IPC. On the right is we do the same experiment using the albumin, okay? So you can see that if with 100% albumin, pure albumin, the survival rate of the stem cell is a big 0, okay? And you need to keep on diluting the plasma with our formula to almost 1/3, 2/3 before you can see them to increase. And you can also see the dot on the 10 right here is actually the dot on the left here. So you can see the albumin curve actually goes down like this, and which is very different from the synergistic effect we can create with human plasma and the plasma conditioning formulations. So with this finding, we know you cannot use only albumin to accomplish this type of results. So in conclusion, plasma is actually very important for the functional stem cells, and the conditional plasma is very important for repair capacity because it is important for with the stem cells. So those were preliminary preparations before we engineer a more clinical program. So we now separate the practice into two phases. One is the intense phase, and the other one is the maintenance phase. So for the intense phase, you do it every 2 weeks, and you do it 3 times. So with this, you will exchange about 450 cc of plasma. And after that, you can change the interval of the plasma in between the plasma conditioning from 3 months to 6 months. And the volume can also decrease a little bit if you think the person is doing well and you don't need to do 150 cc plasma conditioning. In between, we do sometimes give patients some hormonal or cytokine supplementation on a shot basis. Basically, we only give them, say, for 1 week or 2 weeks. So Mr. Lu had asked me to go into more details about the report we will send out from the plasma diagnosis test. So this is the report of from cover to cover interpretation, okay? The interpretation. So the first thing is the outline, then you see the basic data on the patient. And this is the gross appearance of patient's blood. Then you can see on the right, you can see some of the related anti-aging related index, which we do look at for the plasma conditioning. Basically, we look at the reticulocyte count. This is based on we want to use this to look at the bone marrow function. And CRP, you know, is for inflammations. IGF-1 is for the growth factor. EGFR is the renal function, and the hematocrit, that's to prevent anemia. Then after that, you will see this graphic on the left side. So there are 3 colored dots here. So the one right here, normalized to 1, is the original number of the stem cell given to the human plasma. So the survival rate will be of the 100% human plasma will be B prime divided by B. So that's how you get the stem cell viability. So then we look at the right side, then we dilute the plasma by 50% without adding any formulations. If you can, in this case, you can see the viability of the stem cells rises from B prime to C. So this is an indication that the patient's plasma is toxic to the stem cells. And if you dilute the plasma with 50% of formulation, then you can see the stem cell viability has room to grow 1.5 to 2.5, okay? So that is the aim of the target for the plasma conditioning. And this is how you see the data. And on the right side, these are the biochemical reagents. And of course, all of them are pharmacologically licensed, GMP grade since then, and legal approved by the local FDA. And these are the reagents and we use to compound the plasma, the artificial plasma replacement fluid. And these are the photos from the stem cell culture, and these are the comments, and this is the signature by me after I reviewed it. So if the patient has done this for several times, then we do plot the stem cell viability against the treatment for the client, okay? And we also plot the plasma toxicity as well. And the rest of the report are just standard dictionaries and the references, okay? So now I'll quickly show you some clinical examples, and these are quite complete and interesting cases. So this is a guy who has been getting this plasma conditioning therapy in our clinics, Taipei clinic, for about 2 years now, 2 or 3 years, I don't know. And originally he had a stroke and he can't walk at all, and he's recovered now and he can play tennis, okay? So we have a patient coming in for diabetic chronic kidney disease, and she got this therapy, and very quickly, in about 5 months, the eGFR was able to improve by about 15%. And this is a 95 years old lady, and she was one of our original patients, and you can see the results was very good. The stem cell survival rate was improved dramatically with this plasma conditioning. And she also grew out some black hair right here. And this part, the end of this, the hair is actually from the dying of the hair. So, and there are many, many more clinical examples. And I think because of time, we won't go into details. So what we know about this, I want to summarize what we know about the heterochronic plasma conditioning technology right now. Basically, the heterochronic parabiosis was an animal experiment. And for 100 years, people are trying to translate this technology into clinical aging reversal practice, okay? And to the best of our understanding, right now, there are 4 ways you can do this. So the first way is the most natural way. And how you do it is you can remove the aging substance by menstruations, and you can add in the useful substance by getting pregnant, okay? And the other way is a biological way. This is what the Ambrosia, the American company, does. You can do bloodletting and replace them with FFP. And the physical way are people who practice TPE, plasmapheresis. You can remove the toxic substance or by using the machine. And again, you can use different plasma replacement products. And what we are doing is we use bloodletting and some formulation to remove the aging substance, and we replace them with a proprietary combination of biochemical nutrients and the reagents. And there are pros, cons for different approach. And the natural way, the pregnancy way, is free, but is most expensive in the long run. And the biological way is limited by its supply. And the physical way of removing the aging substance by the machine, I think, is a bit dangerous. The complication rate is a little bit high for our like, and the psychological effect and the cost of the machine and the HR issue and the labors are quite high. And another drawback for this is the effects actually only lasted for about less than a month. And lastly, our method has some advantage because it's a mild and long-lasting procedure. But the setback is it requires multiple clinical visits. And the effect is not as fast as the physical way of doing this. And the results are progressive, okay? And some of you may be interested want to know some clinical trial data. And you are all familiar with the classification of the level of evidence in this graph. And at the lowest level, that's based on the case report, you can see the various effect of the plasma conditioning on our method. So most of the clients become more energetic and they don't catch cold, and the control of the diabetic usually gets much better, and their cognitive function sometimes can improve dramatically. We had just one case whose MMSE score was like 11 or 12 out of 15. And by doing this plasma conditioning 3 times in a week, because the patient flew in from Singapore, and both couple improved from 12 to 15 out of 15 just after 3 sessions. And the renal function, especially for diabetic chronic renal disease, we can definitely improve them. And some of the cancer patients coming in after chemo and or major surgical resections, and by getting this, they will become much healthier, and they also become much more energetic. And in some extreme cases, they were able to extend their survival lifespan for up to an year for stage 4 pancreatic cancers. And these are next level of evidence that shows the effect on the renal disease. And these are small trial on the immune functions and the thymus function using similar technologies. And this is the 3A data on Alzheimer. And the number of the patients in this study is about 500. Okay, but with all those small scale clinical trials, I think you probably all understood it is very difficult to conduct an anti-aging clinical trial because of the nature of this issue. Just like in plastic surgery, it's very difficult to conduct a clinical trial because you cannot do a double facelift on one side and leave the other side untouched, and the patient, the control group will not stand this, okay? So, but I think the most important clinical trial data, which is the antidote retrospective study on the effect of pregnancy on the mother. Like what I just said in the beginning, the mother is usually, at least say like 20 to 45 years old, older than the fetus. And they were connected for say like 10 months. And especially for the last 3 months of the pregnancy, the fetus grow to a significant size that matches about 1/30 or 20th of the weight of the mother. So pregnancy is a very good clinical trial of the effect of heterochronic parabiosis on longevity. And the data here shows if the patient, the mother, has her last pregnancy at age of 45, that's shown on the dashed line, and the probability of her to live to age of 80 is 50% higher than the mother who has the last pregnancy of age 35, because the effect of heterochronic parabiosis is much bigger in the first prior case as compared to the latter case. There are also many potential applications, and which is being proposed by Thomas Randall, who is responsible for the resurrection of this century-old technology in the studies of anti-aging. And there are also different clinical trials of using different plasma formulations in Alzheimer's disease, in Parkinson's disease, and things like that. And we think within 3 or 4 years, we should have some medication, IV infusions, which has the effect of reversing some chronic disease approved by the FDA. So the plasma condition therapy, you can use this independently to activate the resident stem cells. And you can also use this as a pre-treatment before you do the cell therapy, okay? And I think this is the position you can consider for clinicians to incorporate this technology into your practice. So, because if you infuse the stem cells without the plasma conditioning, sometimes you will run into a situation like the left-hand side. That means that you bought 100 stem cells infused into a body, but only 3 of them survived. And unfortunately, this is most of the time, this is the case. And what is even worse is that the 97 cells which don't survive actually will bring a lot of free radicals and the toxic substance and release them into the bloodstream. So a lot of the patient who doesn't feel quite well or have a feverish response is actually due to the death of the stem cells infused. So with our technology and the plasma and the stem cell viability testing, you can find the best window to perform the stem cell transplantation, and the patient will get much better response with less dosage. Okay. So in the future, and we are expanding this type of technology into intravascular stem cells. Okay. And these are 2 types. One is the EPC, the other one are the NK cells. And we should be able to offer this type of plasma conditioning by the end of this year. And we are also begin to bring in more biochemical reagents and trying to extend the efficacy of the therapy. And we also set up different plasma conditioning laboratory and the clinics in the China and the Asian area. And we are very open to collaborate with people in every city. And if you are interested, please contact us. And this is our plan for 2023. And this is, we're interested in setting up a center in Jakarta, Ho Chi Minh City, Singapore, Kuala Lumpur. This one is coming up very soon. And Phnom Penh, Bangkok. And these are the cities in China. And we are also in the talk of setting up our flagship center in Tokyo, in the Omotesando area. There is a very important hurdle we need to overcome in performing the plasma conditioning. Is because of the human error, because these technicians who is required to perform such a program is very high. So with that in mind, we cooperate with MIH company in Japan. We already developed some automated plasma conditioning and compounding device, and who are able to do the the stem cell viability test and the compounding the artificial fluid for the plasma conditioning automatically to reduce the probability of error. So this is what we do now. Basically, we draw the blood from the patient and we give it to the machine. The machine will come up with a report, and the report, after approved by the doctor, and will give it to the pharmacist, and the pharmacist will bring the the drugs and the IV FDA approved fluid into the machine, and the machine will compound the artificial plasma replacement fluid for that individual. Okay. So stem cell dysfunction is the cause of aging. And the old plasma is definitely not good for the stem cell function. So intermittent plasma conditioning technology can improve the stem cell in the old plasma. And that's the reason, and the results can last for several months. And that's the reason we think heterochronic parabiosis based anti-aging plasma conditioning is the way to go for the anti-aging market in the future. So this is we are coming to the end of this talk, and let me just tell you a little bit more about some of the new things which is coming up. So this is an experiment done by the Columbia group, and what they do is that they are able to using the injured tissue, injured organ. In this case, is the injured pig lung of the pig, and it connected to the another pig. Okay, and you can see that the injured lung can recover in about 36 hours. Okay, and this type of technology is called cross circulation organ restoration technology, and we are also working on this. And we are using a rat model, and we are working on this using the AKI animal model, and we can see that the kidney, the injured kidney of the AKI, can be improved very quickly. And what comes to is almost like a totally surprise is this patient I show you we discussed a lot previously during the talk, is actually a 93 or 4 years old guy. Interesting is this guy went to States to visit his daughter and his son-in-law, and he had suffered a fall in the AKI, and the kidney function was gone. And for his age, he was 95 years old, and in 1 month, his renal function was fully recovered. Yeah, and because this person has been getting this our plasma conditioning program for 2 years, and I think the family is totally grateful about this. So these are the machines we are designing, and hopefully in the future we will be able to use this machine to restore the function of various organs. And Professor Oka was my mentor in the Department of Chemistry at University of Chicago, and he always tell us this. Trying to just go ahead with what you think is right. And in the beginning, people is going to think it's impossible. And after you get some results, and people will become skeptical. And once your results has gained popularities, and then the people will say that it was obvious. And I think we are at the stage of between the skeptical and the obvious. And based on Dr. Oka's opinion, the first 3 years has passed, and we are trying to use the next few decades to establish the efficacy and the evidence and the clinical data on the heterochronic plasma conditioning technology. And with that, thank you for your time and the listening. I'm open for questions now.
Thank you, Dr. Pan. I think today's sharing and presentation is wonderful and very comprehensive. Should we go for a 2 minutes break, Dr. Pan, or should we just carry ahead?
Yeah.
Okay, then I think we'll just go carry ahead. So now we enter the Q&A session. So for any audience that would like to raise any question, any query, please take a think about it, and the floor is open now. You can just unmute yourself, unmute your mic.
On the mic, I think it's on the left lower hand, I think. I'm not sure.
Right. Dr. Nora, Dr. Amin, or Alice.
What? Who's asking first?
Yes, please go ahead.
Well, I'm Dr. Nora. I just want to ask the stem cells that you actually advise to use. Is there a particular amount? Because you said the lower the better. Because generally, what's been advised to use is about 100 million to even up to 200 million per session, and depending on the ability of the patient to purchase it. But what is your advice? If the patient has already gone through the conditioning, plasma conditioning, then what is the amount that should be used?
I used to use 100 million in the beginning, okay? And with this technology coming up, I mean, I'm using less than this. Sometimes I use 5 million now, and I think I can, and the patient feel good and no side effects. Yeah, you can go as low as 5 million, or if you want, you can try 1 million. But it really depends on the result of the stem cell viability test. If the patient can grow the stem cell, all you need to do is give a little bit, and they can grow 100 times more stem cells. And these stem cells, because these are MSCs, so the size of these MSCs is about 40 microns. And since the capillary of the lung is about 8 microns, so those MSC infused will be in the lung, will be stuck in the lung. And these MSC, if they survive, they will be send out signals, cellular signals based on what they detect in the bloodstream. So this is the way we practice stem cell therapy now. We just use less and less stem cells. And for the patients, they are happy because stem cells are quite expensive, and they are willing to transfer their budget for the stem cell therapy and for the plasma conditioning. And most of the time, actually, they feel pretty comfortable about this type of arrangement.
Okay, so Dr. Pan, so I'd like to repeat what you've just said in your lecture. Ideally, we do conditioning, plasma transfusion, at least about 3 or 6 times before you actually start the stem cells, the combined stem cells.
Yes.
And initially you can do, depending on time constraint, they can even do like every other day, 3 times a week. Or if not, you do twice a week.
Yes.
And on the sixth, after the 6th session, then you put in the stem cells for better results.
Yes. Yes.
And but when do we do the test to assess the improvement in the stem cells once it's injected? I mean, at the beginning of when you do the plasma, or when you actually infusing the stem cells, and then to do it again 3 months later?
We do the plasma, the stem cell viability test before the plasma conditioning, and 2 weeks after the plasma conditioning, before the stem cell infusion. Okay. So basically, we do the stem cell viability test first. Then we do the plasma conditioning and wait for 2 weeks. Then we repeat the stem cell viability test again. So that can document the improvement of the human plasma in terms of the stem cell viability. And after that, since the stem cell the viability already improved, then you can infuse the stem cell into the vein.
Then after you infuse the stem cells, do you advise also to do the stem cells viability after 6 months to see how much is viable at that stage before you say you want to repeat another dose of stem cells?
The stem cell viability test, what we are doing is a test for the quality of the plasma. So you can do, so basically we are testing the plasma, the quality and the bioactivity and the toxicity of the plasma with respect to a certain type of stem cells. Okay, so after you infuse the stem cells, it's really, I mean, you can do another plasma viability stem cell viability test in maybe 3 months. That's what we usually do, 3 months interval for plasma for stem cell viability test.
Say in a person who wants to stay young, forever young. So what's the number of times a patient should have stem cells annually? How many times a year?
It depends on the it depends on the stem cell viability test. If the stem cell can grow in the plasma, they only need to do it, say, annually, or sometimes they don't even have to do the stem cells. Once the quality of the plasma improves, all the stem cell original, we call it resident stem cells in each organ will be activated. But it's really up to different physicians and the different patients' situation. So it's no one answer for this question. Yeah. My advice is that you do have to do stem cell viability test before each stem cell infusions, and I think that's what patients like to see. Yeah.
Thank you for the answer.
Thank you, Dr. Nora. Is there any other audience here like to have another questions?
Good morning, Dr. Pan.
Yes.
I'm from Surabaya, Indonesia. So I have a question. So we already received about the regulatory requirements, and it's written that the active ingredients of these products will keep as a secret. So yeah, to keep the intellectual and competitiveness for this industrial. So I want to ask, in Indonesia, if you want to open a clinic for this, we have to receive the approval from the government, and we have like a Indonesian Doctors Association, and we also have to receive the approval from them. So it means so they have to know about the ingredients, about the bioactive from this product. So what's the solution of this?
So it's pretty simple, because if you want to do a clinical trial on this, you need to, basically this is the reason I try to tell you about what Columbia University did for the organ restoration, the xenogenic cross circulation, because you can create a formula X. Okay, that's the US FDA approach, because so many situations like this, because each people has a new formulations. So you can use a combo combination of the materials and giving it a code, say like 601A3, things like that. And you can apply for a new drug testing with this type of approach. Yeah. Is that clear?
Okay, thank you so much, doctor, for the answer.
I don't know about the situation in Indonesia. I'm sorry. Yeah. But I will be happy to learn more about it. Yeah.
So it means that we just do the clinical trial for the products, right, in Indonesia?
Yeah, yeah, that's correct.
Okay,
just like
thank
you
you.
do a new drug trial. Yeah, yeah.
Okay, thank you so much, doctor, for the answer.
You're welcome. Well, anyway, the first time I think we have an audience from Indonesia.
Right.
Dr. Pan, can I ask you a question?
Of course.
Yeah, Dr. Amin, I'm using Dr. Nora's phone here. I don't know whether it's related or not. It is regarding the question of my patient, whether you can detect spike protein in your circulation. And if it is possible, then through the what do you call this, plasmapheresis for removal, and then doing your plasma reconditioning, does it have any effect at all?
No, you can do many things because what we are talking about today is, I mean, I think every doctor has a secret technology and the ingredients, and that's fully respected. So what we are trying to do is we're providing a stem cell viability test, okay? So right now we're going to do, we are using the extravascular stem cell, and we are going to offer intravascular stem cell viability test by the end of this year. So you can, using our technology as assistant for you and incorporate it into your program, which you think is good. And because every place is different, because in China where I am right now, the customer seems to be worrying about removing blood more than go on the machine. I'm not sure about that. And maybe due to their marketing skill or something like that. But anyway, every place is different. And that's what I say, there are 4 approaches. One is the natural approach. And of course, that's not related to the doctor. And then you can do the FFP, the biological approach. You can combine it with the physical approach with the machine. And you can also using our formulations. But it doesn't matter how you approach this. Yeah, I mean, I think our stem cell viability test is going to give you a very good support and to convince your customer about what you do.
Okay. Okay. Thank you. Thank you. Yeah.
Sure.
Hi, Dr. Penn, it's Alice.
Hi, Alice.
A quick question, I think, I've been sort of following your work for many, many years. I think this is just, I think another step in exciting sort of evolution or improvement with the biochemical test that we are able to do. So now if you're, sort of supplying the plasma to individual, let's say clinics or collaborators. So is there a lab or something that they're going to establish so that these people, because whatever data that you have is quite unique. I don't think every lab will be able to produce the kind of a test that you're able to, to comment on. So is that going to be a lab established in Kuala Lumpur or something that we are going to be able to send the blood, patient's blood for interpretation and all that?
Yeah, basically what we do now is this. In the clinic, in the clinic we collaborate with, you can do we just send our technicians, we call it satellite clinic, the technician from the satellite clinic to collect the blood. And they will process the plasma in the satellite lab. Then we send it to a regional lab. So in Kuala Lumpur, we are setting up the regional lab, which can do the stem cell viability test, okay? But the data interpretation and the final compounding calculations, that's centralized. Is that clear?
Okay, centralized, right?
Yeah.
So
the calculation part and the interpretation, because it's centralized. So the interpretation should be in good hands. Yeah.
Okay. So centralized means the blood is sent to Taiwan, or it's going to be
you do the test in the local. You can send your plasma, say you can process in your, say for example, you want to do it in your clinic, just call us. We send somebody to come and pick up the stem cell, pick up the plasma and go do the test.
So the final formulation is centralized, means it's going to be, you will be the one looking through all the reports and then formulate a certain compound the plasma. Okay. Yeah. All right. Okay,
I'm just overseeing this, make sure that I'll be the co-signer of all the results. Yeah.
Okay. All right. Okay, thank you.
Sure.
Thank you, Dr. Alice. Yes, our lab in KL will be opening soon and hopefully can be operated in the month of July. Yeah, I'll update every one of you. So, do we still have any other questions? Otherwise, we're going to end the Q&A session.
Dr. Pan, can I ask again about the, you mentioned about the, can you hear me?
Yes.
Yeah. So to touch on your experience on the cytokine storm, what was it like, and is there any anticipation as to the possible patient and the early development of the cytokine storm?
Yeah, because when we had one suspicious case about that, yeah, and at that time we don't have that much knowledge about it, but since the COVID-19 is so much in the beginning of COVID-19, there are so many cytokine storms. So I mean, it's basically we are feeling more comfortable about the type of situation. But even we can manage the cytokine storm, with current hydration since all those therapies. But our formulation has going to 4th generation now, so the risk of allergy has decreased by about 10 times, and we have not seen any cytokine storm anymore. Yeah.
And just now, if I get it right, in terms of the maintenance therapy, the recommendation of the intermittent injection of interleukin, how is that being done?
Okay, so if you want to go into maintenance therapy, because some of the components in the plasma, the artificial fluid is too short, so you can do some hormone and some cytokine combinations, doing a subcutaneous injections, like small shots. Yeah, we also compounding those material for you.
But that is optional.
Yeah, that's optional. Yeah.
Okay, thank you.
All right, well.
Before that, just one more question.
Okay.
I just want to find out regarding your your stroke, with patient with stroke. Does it matter that if you receive a treatment in the early on, immediately after the stroke, or if patient already had stroke like 2 years down the line, do you think that you would still be beneficial to receive plasma conditioning, and that you will probably still expect results?
Yes, definitely, because I mean, if you're just trying to be very critical, looking at what we are doing, basically we are changing the intravascular volume, the proportion of all materials in the intravascular, and since vascular, the quality of the intravascular environment is related to all the regenerative procedures and all the circulation and things like that. You can name 100 of those effects. So almost everybody, after they got the plasma conditioning session, they immediately feel relaxed, or they feel just energetic and things like that. The issue is right now is what you're asking about the 2 years down the road. I think, even for people never have a stroke, they're going to feel the similar improvement. So for people who had stroke 2 years ago, I mean, they still experience, it's still beneficial to them. Yeah.
Okay, thank you.
Well, okay. For that, I think we are going to end today's sessions. Thank you, Dr. Pan, and all the audience and delegates who make time to attend, make our event today successful.
Okay.
I'll end the floor today now. Thank you. Bye-bye.
Thank you, Dr. Pan.
Bye-bye.
Bye.